Assalamu alaikum guys, welcome back to another video by Science with Kaswani. Today we are gonna be solving Feb March 2026 paper 12 for A level biology. So let's get right on with it. So we have a eyepiece graticule, and the question is saying that the eyepiece graticule for a light microscope at 100 magnification, or you could say 100x magnification, was calibrated using a stage micrometer scale as shown in the diagram. All right, so you know this by now. Whenever we talk about a light microscope, we talk about two scales, right? Eyepiece graticule and then stage micrometer. Eyepiece graticule is the scale which is inside of the microscope. So you know that thing, that the piece in the microscope which you put your eye into in order to use the microscope. The eyepiece graticule is inside that piece or that part of the microscope. However, a stage micrometer is not really part of a microscope. You put it on the microscopic slide. So, whenever you gave your P3 exams or when you performed your lab experiments, you must have been given a slide, right? And you place that on the microscope and then you just look into the microscope to see what is it. There is a special type of this slide which is called a stage micrometer. So normally this glass sort of dish or this glass sort of slit normally contains, let's say, a dicot stem or a monocot stem or anything which you have to analyze and draw, etc., etc., right? There are some special glass slits which have scales printed on them, and these are the stage micrometers. So, what you're doing is you're calibrating the eyepiece graticule using the stage micrometer. Now, how do you calibrate an eyepiece graticule? Well, here's how we do it. Each small division on the stage micrometer scale measured exactly 0.01 millimeters. So each small division, each small scale which you see here in the stage micrometer, this is exactly zero point zero 0.01 millimeters. So let's say if this is 0, then this is 0.01, 0.02, 0.03, zero point zero 0.04, this is 0.05, and then 0.6, 0.7, 0.8, 0.9, and then eventually this is going to be zero point 0.1 millimeters, so zero point 0.2 and zero point 0.3 millimeters, and so on and so forth. What you have to do is compare the number of divisions in the stage micrometer and in the eyepiece graticule. How do we do that? Let's take an example. Let me just erase all of these values. Okay, let's see. If you take a look at the end of these two scales, right? The line or the place at which these two scales end, they end at the same line, right? Now, right here, I see the number 100 on the eyepiece graticule. What is this number 100 telling me? What does this number 100 stand for? This basically stands for the number of divisions. Look at the eyepiece graticule. One division, two, three, four, five, six, seven, eight, nine, 10, right? So 10 divisions. Most students get confused by thinking that, oh, 10 might mean 10 micrometer or 10 millimeter. No. In an eyepiece graticule, the number is not a length. It's not 10 millimeters or 10 centimeters or 10 micrometers or 10 nanometers. It's not length. It's divisions. It's telling you the number of divisions. But this is not the case in a stage micrometer. There it's talking about length and not divisions. So, can we say that 100 divisions on the eyepiece graticule is actually just equal to this much of a stage micrometer? Because it ends on the same line and it starts at the same line as well, right? So you can say that 100 eyepiece graticule units, I'm writing eyepiece graticule in short form, is equal to how many millimeters? If this is 0.01, 0.2, 0.02, 0.03, 0.04, 0.05, 0.6, 0.7, 0.8, 0.9, 1. So this is 0.1, 0.2, 0.3, 0.31, 32, 3T, 34. 0.34 millimeters. So in that case, one eyepiece graticule unit is going to be equal to how many millimeters? Simple, just use the ratio method. Ratios are your best friend. So this basically just becomes x is equal to 0, just cross multiply, right? So this basically becomes 0.34 divided by 100, which is equal to, I don't know, I'm just going to use my calculator just to be extra safe. So 0.0034 millimeters. What is this? This is what one small eyepiece graticule division, the smallest division, is equal to 0.0034 millimeters. Now, a single celled organism was viewed with a light microscope. The length of the organism measured 47 units on the eyepiece graticule. You know that one eyepiece graticule unit is equal to 0.0034 millimeters. So, how many millimeters is going to be 47 eyepiece graticule units? So, x millimeters. And that is all that you have to find. So, 0.0034, multiply this by 47 and you get 0.1598 millimeter. All right, now here's the time where you convert your answer to such a unit which is being used in the answer choices, right? So they're using micrometers. Now, the way I teach my students of how to go from millimeters to micrometers, now I know there is an easier method to this, but this is just how I teach it. Whatever unit you're given, whatever, no matter what it is, always convert it to meters first. So, in this case, you're given millimeters, convert this to meters. And then once you have it at meters, now you can convert it to micrometers. Now, I know some of you might be thinking, why can't we just go from millimeters to micrometers? You can. But in that case, you're going to have to remember how much do you multiply it by. Now, let me ask you a question. What if instead of millimeters, this was centimeters? Then you would have to remember a different number, right? As to what number would you use to go from centimeter to micrometer? Do you multiply by 100? Do you multiply by 1,000, 10,000, 100,000? So if it's millimeter, you remember a different number. If it's centimeters, you remember a different number. Why do we need to remember so many different numbers? Just remember one number, and that is how to go from meters to micrometers. And that is multiply by 10 to the power of 6. Simple as that. Now, you do know how to go from millimeters to meters, right? Millimeters to centimeters is divided by 10, centimeters to meters is divided by 100. So, in other words, if this is divided by 10, this is divided by 100, this is overall divided by 1000. So 0.1598 millimeters when divided by 1000 becomes equal to 1.598 times 10 to the power minus 4 meters. And now your answer is already stored on the calculator. Just press the multiplication sign, 10 to the power 6, because now you're going to convert the meters to micrometers, and this becomes 159.8 micrometers. Or you could say 160 micrometers if we just round it to two significant figures. And that's our answer right there. Question number two. One of the electron micrographs shows a eukaryotic cell. The other shows a prokaryotic cell. What is the approximate distance across the eukaryotic cell at its widest point? Now, this question is a bit interesting in the sense that they haven't given you much information, right? And some students are going to be confused on how to solve this. But trust me, this is pretty simple. Your first step is to identify which is eukaryotic and which is prokaryotic. And this is pretty simple, right? We know that prokaryotic cells don't have a nucleus. They instead have a nucleoid region. They don't have a true nucleus, or a true nucleus basically means a nucleus which is surrounded by a membrane. This right here is the small nuclei region. This does not have a membrane around it, right? Normally, in other pictures, in other diagrams, nucleus looks something like this, right? Which is exactly what we see right here. So, because we do not see a well defined or a true nucleus here, this is a prokaryotic cell, this is a eukaryotic cell. Right. Step number one is done. So, this is prokaryotic and this is eukaryotic. Now, what you're going to do in the exam is you're going to take your ruler and notice the entire diagram does not say anywhere not to scale, which sometimes it does say. Because it hasn't mentioned it here, the examiner is trying to give you a hint that yes, you can use your ruler on these diagrams. Now, how do you use your ruler in this case? Look at the question again. It asked us the approximate distance across the eukaryotic cell. So this is out of the picture at its widest point. So take a look at this eukaryotic cell. What are the two farthest points away from each other? Or what are the 2 points that are the farthest away from each other? Is it this or this? No. Is it this or this? Nope. This or this? Nope. What about this and this? Yes. So now you put one end of your ruler here, the other end of your ruler here, and then you measure this length. Think about it. This length which you're measuring this in the image, right? You're not measuring this in real life. So the length which you're gonna get, is it gonna be the image length or is it gonna be the actual length? Of course, it is gonna be the image length. Now you have the image length and you have the magnification. You know the formula. Magnification is image size over actual size. So actual size becomes image size over magnification. You got your image size, put that in there. You have your magnification, put that in there, which is 4000, and you got your actual size. I can't measure it here because it's on an iPad screen, but if you, let's say, somehow, hopefully, because this is a pretty easy question, if you get it in the exam, just measure it, put your answer here, put the 4000 here, and then you get your actual value right here. All right. Sorry. I'm just a bit sick, so you might hear me coughing again and again. So, really sorry for that. Okay, question number three. Which polymer is a major component of the cell walls of typical prokaryotes? Now, whenever you talk about cell walls, right, there are three names which you should remember and that should come to your mind whenever you talk about cell walls. What are these three names? Number one, the cell walls in plant cells are made up of, everyone knows this, cellulose, right? That's number one. Number two, the cell walls in fungi. Remember that other kingdom? Plants, animals, fungi, bacteria, protocosts. So fungi is another kingdom which also have cell walls. Their cell walls are made up of a substance called chitin. Make sure to remember this. This also sometimes comes up in some MCQs. And finally, if you talk about the cell walls of prokaryotes, or in other words, bacterial cells, so I'm going to write prokaryotes slash bacteria, then these are made of a substance called peptidoglycan. Now, if you don't want to remember such a big name, that's fine. I'm not going to judge you. There's another name for you which you can remember, Meurin. Meurin. Completely up to you, whichever one you want to remember. You are never going to get an MCQ which is asking you the cell wall. What is the stuff present in the cell wall in prokaryotes? And this is option number one, and this is option number two. No chance. Because these two words are the exact same thing, it's just different synonyms of the same thing. So, which polymer is a major component of the cell walls of typical prokaryotes? Peptidoglycan. Simple as that. Question 4. Which features are shown by prokaryotic cells and also by eukaryotic cells? Do prokaryotic cells have a cytoplasm? Of course they do. Think of a bacterial cell, right? The best example of prokaryotes you can remember is bacteria. And the best example for eukaryotic you can remember are actually two. That's animal cells and plant cells. Now, why do I ask you to remember two? Because think about it, right? If let's say you only remember, I don't know, animal cells, and now this is the only one you have remembered, you have completely forgotten about plant cells, and the question is asking you what features are shown by prokaryotic and by eukaryotic. Now, you might be thinking, oh, bacteria has cell wall, but animal cells don't have cell wall. So, cell wall is not a feature that's common in eukaryotic cells. That's wrong, right? Eukaryotic cells are also plant cells, and plant cells do have a cell wall. So basically, what I'm trying to explain is whenever you just think about eukaryotic cells, just think about both of these types of cells together, not just animal cells alone and not just plant cells alone, unless it specifies which type of eukaryotic cells they are talking about. So, Which features are shown by prokaryotic and also by eukaryotic? Do both of these types of cells have a cytoplasm? Absolutely, they do have a cytoplasm. So eliminate all of the options which do not have the number 3 in them, right? This is process of elimination, a very important skill that you should know. So 3 is not in D, so D is cut out. Okay. Now, do they have 70 S ribosomes? If you have learned your syllabus properly, you should know that prokaryotic cells do have 70 S ribosomes. Eukaryotic cells have 80 S ribosomes. But these 80 S ribosomes are in the cytoplasm of eukaryotic cells. But remember, eukaryotic cells also have mitochondria. And if you're looking at plant cells, they also have, on top of mitochondria, they have chloroplasts as well. And both mitochondria and chloroplast have 70 S ribosomes. Now, how are you supposed to remember this? Go to your book and understand the endosymbiont theory. This is not going to come up in the exam, but this is going to help you remember the fact that because mitochondria and chloroplast contain 70S ribosomes, and mitochondria and chloroplast are part of eukaryotic cells. So you can technically say, right, that eukaryotic cells also contain 70S ribosomes. So these have 70S, these also have 70S. So 4 is also correct. So C is wrong too. Now, take a look at A and B. 1, 3, 4, 1, 2, 3, and 4. So it's clear that 1 is also correct. And we know this. This is basic, right? Cell membrane is a feature of a living organism. Now, what do I mean by that? Whenever we use the word feature of a living organism, it means that an organism cannot exist. It's not possible. It's not logically possible if their cells don't have a cell membrane. So it must be present. Are these your? You should ask yourself this: Is prokaryotic cell a living cell? Yes. Is eukaryotic cell a living cell? Yes. All living cells must have a cell surface membrane. So 1 is also correct. And 3 is also correct. 4 is mentioned in both of these, which we also said is correct. The only decision you have to make is lysosomes present in prokaryotic cells and eukaryotic cells, or in either one of those? So think about it. You know that lysosomes are in eukaryotic, right? We've learned all about them. They are digestive, they contain digestive enzymes, and they secrete those digestive enzymes to digest a cell, blah, blah, blah. So these have lysosomes. But then what you should remember is prokaryotic cells do not have lysosomes. So 2 is wrong. So B is our answer right here. Okay. Some proteins synthesized in the plant cells are modified before they are secreted. Which cell structure in the plants is responsible for modifying the proteins before they are secreted? So I'm going to explain the entire process here, and then you guys are going to give me the answer for this question. So what basically happens is inside plant cells, the first step to making a protein molecule, right? The first step happens in the ribosomes. This is why ribosomes are also called the site of protein synthesis. Now, let me ask you this. Could you tell me the two locations in which ribosomes can exist inside a plant cell's cytoplasm? I'm going to repeat. Could you tell me the two locations in which ribosomes can exist in a plant cell's cytoplasm? Now, I'm not talking about inside a mitochondria or inside a chloroplast. No, inside a plant cell cytoplasm. So that's just two. Either they are just freely scattered in the cytoplasm, or they are on the cisternae of the rough endoplasmic reticulum, which we call the rough ER. Right, so this is the cisternae of the raphia, and we have these dot dot structures called ribosomes, obviously, on their cisternae, and this is where protein synthesis occurs. But there's a problem. What's that problem? When the proteins are being made in these ribosomes, right, these proteins which are made, they cannot currently be used. They're not in a state of being used, right? So, for example, Let's say your house is being, I know this is a very unrelated example, but let's understand this. Let's say your house is being renovated, right? And the painter, he puts the first coat of paint on the wall. Is your house ready? No, he just put the first coat of paint on the wall. He's gonna put a second coat, then a third coat, then a fourth coat, blah blah blah. So even though the basic structure of the wall is ready, it's still not complete. Same case here. Even though we have made the primary structure of the polypeptide chain, it's not yet functional. It cannot yet be used. So, how do we make it functional? The thing is, once the proteins are made by these ribosomes, these proteins are then transported. The proteins which are not yet functional, by the way, they are transported in the vesicles to the Golgi body. Now, what's a Golgi body? A Golgi body is pretty similar to an endoplasmic reticulum in the sense that it also has cisternae. Cisternae is just basically these flattened sac-like structures, but they do not have any ribosomes on them. Now, the vesicle, which came from the rough ER, is moving, and then it fuses with the Golgi body. This vesicle contains that unfunctional protein or non-functional protein. Now, because this vesicle has fused with the cisternae of the Golgi body, now this non-functional protein is moving through the Golgi body. Now, here's the important part. While it's moving through the cisternae of the Golgi body, now this is where it is modified. Its structure is modified. Let's say a lipid molecule is added to it, or a glycoprotein is formed because of two lipid molecules being added to it. So different extra things are added to this protein molecule in order to make it completely able to use, or let me rephrase, in order to make it functional. So are we actually just modifying the proteins? Yes, we are. So, which cell structure in plants is responsible for modifying the proteins before they are secreted at the end? Secreted obviously means that if they're being secreted, they're ready to be used, right? So, who's modifying those proteins? It's the Golgi body. Okay. Now, which row shows the types of membrane that surround the mitochondria and the nuclei? This is pretty simple. Both mitochondria and the nuclei and the chloroplast. These are the three, or I should say, the only three organelles, the only three structures inside of the cell which are surrounded by two membranes. So mitochondria and nuclei both are double membrane structures. This is a 5-second question. Don't take more time than needed. Okay, part 7. It is suggested that primitive prokaryotic cells, what does primitive mean? Primitive means old, or you could say ancestral prokaryotic cells. Now, as soon as they have used the word primitive, you can feel right it's going to be related to the endosymbiont theory. But let's see. It is suggested that primitive prokaryotic cells may be, see, ancestors of certain organelles in eukaryotic cells. And we know what organelles are they talking about? Only two of these that are mentioned in the endosymbiont theory. If you have no idea what I'm yapping about, just go look at in your book, the endosymbiont theory. That's mitochondria and chloroplast. So, which organelle is most similar to a typical prokaryote? So just look for mitochondria or chloroplast. There we have it. C is our answer. Now, which of the sugars, fructose, glucose, and maltose, are reducing sugars? Now, let me tell you this something. In your syllabus, the word reducing sugars is not even defined. If you want to understand what reducing sugars actually are, these are basically sugars that act as reducing agents. Now, I know this is not in your syllabus and you must have never learned this, but I'm just going to give you I'm just going to try to give you some sort of a insight on why do we actually use the word reducing. Reducing sugars are called reducing because they act as reducing agents, right? You must have learned this in chemistry. They reduce other substances and they themselves get oxidized. But then what do you have to know for this particular question? This is just something that you should remember. Now, what do you need to remember? Whenever you talk about reducing sugars, you can divide these into two categories: monosaccharides, so basically reducing sugars that are monosaccharides, and reducing sugars that are disaccharides. Now, just remember the sentence which I'm going to write right now. All monosaccharides, all monosaccharides are reducing sugars. Whatever monosaccharides you have learned about, whether it is glucose or fructose, these two are majorly the ones everyone knows about, the most popular ones. These are reducing sugars, right? So, fructose, reducing sugar, glucose, reducing sugar. This is reducing, this is reducing, and then this is also, and then this is also. How about disaccharides? Now, this is where things get a bit interesting. Now, why is that? It's because there are some disaccharides which are reducing sugars and some disaccharides which are not reducing sugars. You have only learned about two disaccharides, that is maltose and then sucrose. Now, this is what you need to remember. Maltose is a reducing sugar. Sucrose is not a reducing sugar. Simple as that. Right. So let's see that we have fructose and glucose only. Only is not the case, right? Maltose we just saw is also reducing sugar. So this is wrong. Again, only no. Fructose, glucose, and maltose. Exactly. And again, glucose and maltose only, not only. Fructose could also be an example in option number D. Okay. Now, question number 9. Which molecule in a muscle cell is rapidly hydrolyzed to produce glucose? What does the word hydrolyze mean? Hydrolyze means breakdown of a molecule. You're breaking down a molecule using water. Which molecule are you breaking down using water to make glucose? Let's see. Amylopectin, then we have amylose, cellulose, and glycogen. Cellulose is the polymer I believe used in plant cells, right? Yes. So you never break down cellulose to make glucose. So absolutely not. This is pretty simple. You just break down glycogen in order to produce glucose. You don't break down amylose. You don't break down amylopectin. Where have you learned these two polymers? These two polymers are part of starch, and starch is present in plant cells and not in a muscle cell. All right, now let's look at question number 10. The diagram shows how the beta glucose monomers of cellulose are linked to each other. The OH groups on carbon number 2. What's carbon number 2? Always remember, whenever you're numbering the number of carbon atoms in a beta glucose molecule, this is how you number. So this is 1. Always start with the one which is just below the oxygen. So this is 1, 2, 3, 4, 5, and 6. But always also remember, in cellulose, every second beta glucose is inverted 180 degrees. So basically, this right here is gonna be the one. So this is 1, 2, 3, 4. Or in fact, 1 second. Let me just double check. Yeah, this is fine. This is 1, 2, 3, 4, 5, and 6. Yeah. So this is gonna be number 2. And you clearly see that the OH groups are on opposite sides, right? So what is it asking us? It's asking us what is the main way in which this arrangement, it's talking about this arrangement of the OH groups on carbon 2 contributes to the strength of the structures made of cellulose. Okay. What purpose or what function do these two OH groups have, and how do they play a role in maintaining the strength, or you could say increasing the strength of the cellulose molecule? And this is pretty simple, right? This right here is one cellulose molecule, and this right here, assume there is another cellulose molecule. So there is an OH group, let's say, of beta glucose in the second cellulose molecule. And now this OH group and this OH group are gonna have hydrogen bonds formed between them. Now assume there was another cellulose molecule on top of the one they have already drawn for us, and there was another beta glucose molecule which had an OH here. Now this OH and this OH are gonna form hydrogen bonds. So you see what's happening basically? It's just these OH groups are forming hydrogen bonds with neighboring, or you could say adjacent OH groups that are present on different cellulose molecules. So the OH groups on both sides of the cellulose molecule, yes, they can form cross links within the same cellulose molecule. No, they're forming cross links with different cellulose molecules, right? They have already formed the crosslinks in the same cellulose molecule. So A is wrong. Yeah, with different molecules through the formation of glycosidic bonds between nearby OH groups. No, you're forming temporary hydrogen bonds or strong hydrogen bonds. So within the same molecule, again the same thing. No, with different molecules, nearby OH groups, hydrogen bonds. Right. So D is our answer. Right. Right. So question 11 says which statement relates the structure of triglyceride molecules to their function? So before even reading the answer choices, we know what triglycerides do and why are they needed in our body. Triglycerides basically, how are they made? They basically have a glycerol molecule here and then three fatty acid chains here. Now, because all of these fatty acid chains or the tails of the fatty acids, you could say, have many carbon hydrogen bonds inside of them and many carbon-carbon bonds inside of them. But we mostly just look at these bonds. What's so special about them? Well, every carbon hydrogen bond, so you see this bond here, which I've highlighted in red, this contains a huge amount of energy. So now when I break this bond, it's going to release a large amount of energy, which is going to be used by cells which need the energy to carry out life processes. So in other words, why are we using triglycerides? For storing energy because they have many carbon hydrogen bonds, right? So triglyceride molecules can function as energy stores, correct? Because they have large number of carbon hydrogen bonds in their fatty acid tails. Exactly. Now why is B wrong though? Let's take a look. Triglyceride molecules can, and I should not have circled A first. Let's read through all of the choices and then we'll circle A. Triglyceride molecules can function as energy stores. Okay, interesting. But see this? Because large amounts of energy are required to hydrolyze ester bonds. If you are, so first things first, you're never breaking the ester bonds. Ester bonds are this, not the bonds between carbon and hydrogen. Ester bonds are the bonds between the fatty acid and the glycerol molecule. If you break those ester bonds, you do not have a triglyceride molecule at the end. The triglyceride molecule is broken, right? So you don't break the ester bonds. Secondly, if large amounts of energy are needed to break the ester bonds, then how is that benefiting the body? The body's energy is being used up, right, to break the bond. So you're not really providing the body with energy. Instead, you're doing the opposite thing. You're doing the wrong thing. You're taking up the body's energy. So this is wrong. Now, triglyceride molecules can provide energy to all cells of the body, correct? Because they are non-polar molecules, correct? That easily dissolve in water. There we go. Wrong. If they are non-polar, how are they water soluble? So C is wrong. Triglyceride molecules can provide energy to all of the cells of the body because they are polar. Nope, nope, nope. D is also wrong. So A is correct. Always go through every single word in the answer choices. And this is specifically correct for bio, this trick or this tip, because they just play with you on those one or two words, right? Now, if you would have been reading this quickly, you might have read this as polar and this is non-polar. And this might have made things a bit different. So always just carefully read through every single word. Right. Question number 12. Membrane fluidity. So it's basically the fluidity of the membrane. How fluid the contents of the membrane are. What are the contents of a cell membrane? We have phospholipid molecules, right? Which make the phospholipid bilayer. And then we have the glycoprotein molecules. And then we also have glycolipid molecules. All of these molecules just move around here and there in their line, right? So if the phospholipid bilayer is like this, then the phospholipid bilayers cannot move here or here, they can move within their line. Same rule applies for glycoproteins and glycolipids, and that's what we call the fluidity of the membrane. Now, this fluidity can be affected by a number of factors, right? You know this. This can be temperature. So basically, the higher the temperature, the more fluid the membrane is. Why is that? Think about it. If you increase the temperature, what's going to happen to the kinetic energy of the phospholipid molecules, glycoproteins, and glycolipids? They're gonna have more kinetic energy, so they're gonna move more faster. The membrane is gonna be more fluid, right? They're gonna have more fluidity. So temperature, you could say, is directly proportional to fluidity. Number two, let me ask you this. You have phospholipid molecules in there, right? And you know that phospholipid molecules have two fatty acid tails. If I tell you that every single phospholipid molecule, just assume for a second that you have a cell membrane, and in this cell membrane, inside every single phospholipid molecule, every single fatty acid chain is unsaturated. What does unsaturated mean? Unsaturated means that the fatty acid tails has carbon-carbon double bonds inside of it. And carbon-carbon double bonds results in kinks in the fatty acid tail. Now, if you're not sure what kinks are, I would recommend revising chapter 4, cell membranes and transport. But let me just quickly explain what kinks mean. So take a look at this. This is a simple fatty acid tail, which is carbon, hydrogen, or let me just draw the bonds. Carbon, hydrogen, carbon, hydrogen, carbon, hydrogen bonds, so on and so forth. However, if I give a double bond here between two carbons, this is no longer going to be a straight line. So, in other words, if a normal saturated fatty acid tail looks like this, a unsaturated fatty acid tail is going to look something like this. And now, if I have multiple carbon-carbon double bonds, each carbon-carbon double bond results in a kink. What is this kink? This is the kink right here. You see the direction changing? So this is the kink. One carbon-carbon double bond, one kink. Now let's say if I have two carbon-carbon double bonds, like this, or not here, because then the carbon's gonna have five bonds. Let's say here. So now take a look at this. Now the tail is going to be looking like this. If the tail has way too many kinks, doesn't that mean that more tails are gonna fit in these spaces made by the kinks? Doesn't that make sense? Let me repeat. If a fatty acid tail has more kinks, how are they going to have more kinks when they are more unsaturated, when they have more carbon-carbon double bonds? Then this, in other words, allows extra space for more tails to fit in, right? So, for example, normally one tail was supposed to fit in, but because this tail has kinks, it allowed for two extra tails to fit in. So, what does that tell us? Basically, this just tells us that the more unsaturated a fatty acid tail is, or you could say, the higher the number of carbon-carbon double bonds it has, the more fluid it is going to be. So, which combination would result in the greatest membrane fluidity? So, greatest temperature, and temperatures has to be the greatest. So A and C is out. And then the percentage of the fatty acid tails that are saturated. Oh, saturated should be the lowest. So obviously, unsaturated would be the highest. So lowest is going to be this. So B is the answer, right? Question 13. The solubility in water of different regions of the cell surface membrane changes with the distance of each region from the center of the membrane. If you read this for the first time, just like me, I have absolutely no idea what this meant. So let's read this again. The solubility in water of different regions of the cell membrane. Okay, now it's starting to make a bit of sense. You're looking at the entire cell surface membrane, and now you're looking at different regions of it. So let's say you're looking at, let's say here in the middle of the cell membrane, then you're at the edge of the cell membrane, then you're at the bottom of the cell membrane. And at all of these different locations, you're measuring the degree of solubility in water. So let's say if you have a molecule or 10 molecules, how many molecules dissolve in each location or in each part of the cell membrane? That's what you're measuring, the solubility in water in the different regions of cell surface membrane. So it's telling us that the solubility changes with the distance from the center of the membrane. So let's say I have a membrane right here, right? Now, of course, inside the membrane, we have the phospholipid bilayer. Why am I drawing a phospholipid molecule like this? Because this is the phosphate head, these are the ester bonds, and then this is the fatty acid tail, these two. So this is the first layer in the phospholipid bilayer. I'm just gonna copy paste this so I don't have to draw the entire thing. Yeah. And then we're just gonna copy paste this entire thing now and we'll rotate it like this. So this is the phospholipid bilayer complete. And this right here is the center, and basically measuring your distance from this center. So think about it. Doesn't the center of the cell membrane have this hydrophobic core because of the hydrophobic or non-polar fatty acid tails? Meaning solubility in the center of the cell membrane is going to be zero. Is it zero here? Solubility? No. So A is wrong, B is wrong, D is completely wrong, and C is correct. And why is C also correct? Because as soon as you move away from the cell membrane, either, you know, how in physics you assign signs or sign conventions, you basically say that if we move forward, that's positive, and if we move downwards, that's negative. So if you move, let's say, 4 nanometers ahead or upwards, so that's plus 4 nanometers. And if you move 4 nanometers downwards, that's minus 4 nanometers. That's why we have the signs here. That's signs is just telling you the direction. Regardless, even if you move up or down, you're reaching towards the edge of the cell membrane, right? Where you have those phosphate heads that are polar. So the solubility is going to be higher the more you move away from the center, which is exactly what C is telling us. Right. Question number 14. Which row about the primary, secondary, and quaternary structure of proteins is correct? So let's see. Primary structure, you know, of the proteins is basically where you're just putting the amino acids together. So it's just the sequence of the amino acids in the protein. So is it just telling you about the number of amino acids? No, it's also telling us in which position which amino acid is gonna be there, right? And not just that, you know, there are 30 amino acids in a polypeptide. It's telling us a lot more than that. So it's not gonna be D, right? So this is wrong. Now, it's the result of translation. So it's telling us the primary structure of a protein is a result of the translation of an mRNA molecule by a ribosome into a chain of amino acids. That's true, right? You can say, yes, that's how you make the primary structure. Okay. Primary structure, is it also the sequence? Yes, the sequence of amino acids that is determined by the codon sequence of an mRNA. mRNA has codons, DNA has triplets, tRNA has anticodons. Always remember this. mRNA is codons, DNA or gene, that's the same thing. Gene is a section of DNA. This is triplets, and then tRNA is anticodons. All three of these are basically just three nucleotide base sequences together or adjacent to each other. So this is also right. Now, the sequence of amino acids or the sequence of amino acids in a protein coded by a DNA molecule. Right, this is also correct. Secondary structure. It is the result of attraction between hydrogen and oxygen atoms, correct? So hydrogen bonds, right? Associated with the peptide bonds. No, this is wrong. The coiling of a chain into of amino acids to form a beta pleated sheet or alpha helix. Exactly. How do they do that? How do they form these either of these two things? Using hydrogen bonds. So the first part of A was correct, but the second part is wrong, right? So overall, this is wrong. D is also wrong. Now let's look at B. This is correct. And always contains at least two different types of polypeptides. Yes, correct. That interact to form a shape of a protein. Exactly. Now, why is part C wrong though? Because it is formed by four polypeptides and an additional reactive group. It's not necessary that it needs to be four polypeptides. Yes, it can be, for example, hemoglobin, and it also has a reactive group, the heme group, but it does not need to be four polypeptide chains. It can even be two. So this is not necessary. This is why B is the only answer which has all three parts that are exactly correct. Right. Question 15. Some RNA molecules called ribozymes, right? Okay. So this is an RNA molecule, can catalyze the reaction. Okay. So they're acting as an enzyme. So they can catalyze reactions in a similar way to enzymes. Many of these ribosomes have other RNA molecules as their substrates, okay, and catalyze reactions that break specific phosphodiester bonds. Another important group of ribozymes form the functional part of ribosomes. Okay. Which statements about ribozymes and enzymes are correct? So binding of the substrate to the active site depends on the complementary pairing of the bases for ribozymes and on the complementary pairing of the R groups for enzymes. Now, this might seem difficult, but it's really not. So let's understand why. Whenever we talk about an enzyme, right, I'm not talking about a ribozyme right now, just a normal enzyme. You know that an enzyme has an active site, and we have the substrate right here. of the active site, and now the substrate just goes into the active site and then forms temporary bonds, right? How does it do that? How does it form temporary hydrogen bonds? Well, look at these molecules, the molecules on the outer surface of the substrate. The R groups of these molecules and the R groups of these amino acids on the active site or found on the outer part of the active site on the enzyme form hydrogen bonds. Let me explain this again. What is an enzyme actually? An enzyme is just a biological catalyst, right? All enzymes are proteins, meaning that this entire thing is a protein molecule. And protein molecules are made up of amino acids. So, meaning this is an amino acid, this is an amino acid, this, all of these are amino acids. I want you to look at the amino acids that are present on the outer surface of the active site. So, this is the active site, this thing. This is the inner surface, and this is the outer surface of the active site. That part of the active site which is going to come into contact with the substrate. Now, the amino acids which make up the outer part or the outer surface of the active site, we know each amino acid has an R group, right? What's the general structure of an amino acid? Hydrogen, R, COOH, and then NH2. So each amino acid has a R group. The way the bonds form is when this amino acids R group and this amino acids R group form hydrogen bonds with each other. And that's what it's telling us. But when you look at a ribosome, that's an RNA molecule, right? And RNA molecules have other RNA molecules as their substrate. So this is not going to be a protein when you're talking about ribosomes, and this is not going to be a protein. This is going to be RNA. This is also RNA. How do nucleic acids join together? Complementary base pairing. So, binding of the substrate to the active site and ribozymes depends on the complementary base pairing and complementary pairing of R groups when we're talking about our classic example of enzymes. So, yes, one is correct. Ribosomes and enzymes can catalyze hydrolytic reactions and condensation reactions. This is simple, right? Condensation reactions are when you eliminate water, and hydrolytic reactions are when you use water. And both of these reactions can easily be catalyzed by a catalyst. So, 2 is also correct. But take a look at 3. For molecules, right? For molecules which are made up of the same number of monomers, okay, the number of possible types of enzyme is greater than the number of possible types of ribosome. What does that actually mean? I am absolutely blown away. What the hell is this? This is a statement which you're gonna say no to just because you don't have enough information, right? We just have this. It's this paragraph is just giving us an intro on what ribozymes are and how are they similar to enzymes. Did they talk about the number of monomers anywhere? Did they talk about the number of possibilities of enzyme types? Did they talk about the number of possibilities of ribozyme types? No. This statement is only given so that the candidate gets confused and immediately selects, oh, I don't know, but it might be right. So let me just click it. No, you don't have enough information. This is wrong. So B is the correct answer here. Because the question said which statements are correct. Are you sure this is correct? Heck, we don't even understand what it means. How can we be sure that this is correct? So B is the answer. Question 16. Some students sketched graphs to show Vmax and Km. What is Vmax? Vmax is basically the point at which the reaction is working at its full capacity, right? So, for example, let's say I have a container in which there is a chemical reaction going on. And in this container, I have 10 enzymes. So, this is 1, then 2, then 3, and then let's say 6, right? So, 6 enzymes in this container. The point the point in time when every 6 of these enzymes are gonna have a substrate inside of them. Does that mean that they're working? If they have a substrate inside of them, it obviously means that an enzyme substrate complex has been formed, right? Meaning they are participating in a chemical reaction, they are catalyzing a chemical reaction, aka they are working. If every single enzyme in the container is working, if is there any more space for an extra reaction to happen? Can any more substrate just come into any one of the enzyme and start a new reaction? No, the reaction is working at its full capacity, right? So, for example, when you go into a restaurant and the owner of the restaurant says, sorry, we're not accepting new customers. Why do they say that? It's because their capacity is full. They have a limited capacity to feed a limited number of people in a given amount of time. When that capacity is full, the owner of the restaurant says, please go to another restaurant. We don't have enough space to let you sit down and have your meal, have your food, right? So the point in time at which the reaction is working at maximum capacity, all the enzymes have filled active site is called Vmax. So this is where the word max comes in. Maximum rate of reaction. So with this, you clearly understand that the y-axis needs to be rate of reaction because Vmax is maximum rate of reaction. And this needs Vmax should be the highest point on the y-axis. So it is the highest point on the y-axis in all four cases. And in all four cases, the y-axis is rate of reaction. Okay. What's Km? KM stands for Michaelis. Excuse my spelling because I have absolutely no idea. But yeah, Michaelis, I'm very weak with spellings. Michaelis Menten constant. What is this Michaelis Menten constant? This is a constant which tells you how much substrate, this is important. This tells you how much of substrates are needed, or you could say how much substrate concentration, that's better, is needed to achieve half Vmax. What's half Vmax? Well, you have the Vmax, right? You have the point in time, you have that reaction rate that cannot be exceeded any further. That's the maximum rate of reaction, the Vmax. Just divide that by 2, half Vmax. How much of substrate do you need? How much of substrate concentration do you need to get half Vmax? To get to that rate of a reaction wherein half of the enzymes have filled active sites. So the rate of reaction is half of maximum, half Vmax. So Km is how much of substrate is needed. So would it make sense if you have Km and the x-axis is enzyme concentration? No, right? It needs to be substrate. And plus, it's on the x-axis, not on the y-axis. Vmax and Km are on different, or you could say opposite axis. So C is our only correct answer, not D, because they are on the same axis, which is wrong, not A and B, because the x-axis is enzyme concentration and not substrate concentration. All right. Question 17. Enzymes can be immobilized in alginate beads. Which row identifies the advantages of using enzymes immobilized in alginate beads compared with using enzymes that are free in solution? Now think about it. If you have enzymes randomly roaming around in the container where the reaction is happening, is it going to be easier to purify the products? Because if you have a container, you have the reactants inside there, then you have the enzymes inside there, and then once these two react together, you have the products there. You have to remove these products or extract these products from the reactants and the enzyme. You have to purify these products, right? And it's going to be difficult because even though the reactants are used up, the enzymes are just freely or randomly moving around surrounding the products. So the product is going to be difficult to purify. But if you think about an advantage of immobilizing it, immobilize basically means not allowing it to move. So if you're not allowing the enzymes to move, the product automatically becomes easier to purify, right? Because the reactants are used up. This is not moving. This is the only thing moving. So you can easily purify it. So this is correct and D is wrong. It is easier to reuse the enzymes when making new batches of the product. Yes, because immobilizing in alginate beads, how does that happen? So let's take an example. I have this tube right here, and this tube basically has many beads. So you see those beads used in necklaces? Think of those as alginate beads, right? It's basically a vertical column or a tube of alginate beads. And now in between these alginate beads, you have the enzyme. Is the enzyme part of the solution? Is the enzyme roaming around with the reactants and the substrates? No, it's sitting in one place. It's not coming into contact apart from when it's reacting with the product and the reactant. So it's easier to reuse it. It does not get contaminated. So, yes, easier to reuse. So, meaning B is also wrong. Enzymes are less likely to denature when heated. This is correct. When you are immobilizing using alginate beads, when you give them heat, majority of the heat is taken up by these alginate beads. So meaning the enzymes are less likely to be denatured because they're not taking up all of the heat, right? Majority is being taken up by the alginate beads. There's only some amount of heat energy that these enzymes unfortunately have to absorb. And because it's so small, they're very less likely to be denatured. So this is also correct. And hence A is the answer. Also, one more thing. It's less about who's absorbing what, as in, I said that the alginate beads are absorbing majority of the heat energy. That's right. But there's also another way of thinking about it. The enzymes are locked at their position, right? They have a specific position and they are locked onto it. They're not being allowed to move even an inch. So if they're not being allowed to move even an inch, it means that their active site is not being allowed to change its shape, right? What is denaturation of an enzyme? Denaturation of an enzyme means that the active site of the enzyme shape is changing. How does the active site's shape change when there is no space for that to be able to happen? This is why whenever you heat it, even though, so imagine if you have enzymes in a solution and enzymes in the immobilized in the alginate beads, and you heat them giving the same amount of heat. Here, there is nothing holding them together. There is nothing locking them into the fixed position. So the active site shape can change. But here, the shape of the active site will not change that much because you're not giving any space and hence you're not allowing for that to happen. So it's less likely to be denatured when heated. That's also another way of thinking about the same thing. Question 18. The diagram represents the arrangement of some of the molecules inside of a cell surface membrane. Labels P and Q identify two regions of one of the molecules. Okay, this is obviously a phospholipid molecule, and labels R and S identify two regions of another molecule. This is probably a glycoprotein or a glycolipid. Which labeled regions always, this is important, the word always contain oxygen, and which labeled regions always contain phosphorus? Think about it. In a phospholipid molecule, this is what it looks like, right? So you have this phosphate head right here, and this is the polar phosphate head, and the formula for phosphate is PO43 minus. And then you have these fatty acid tails, which consist of either carbon-carbon single bond or carbon-carbon double bond or carbon-hydrogen single bonds. So phosphate heads obviously contain phosphorus. So P does contain phosphorus. But what about S? S is a receptor. How are we so sure that the receptor is gonna contain phosphorus? Now, I'm not saying it won't, it may, but will it always contain phosphorus? No. So S is not under always, it's not categorized under always containing phosphorus. So A and D is wrong for that specific reason. Now, always contain oxygen. P is PO4. Obviously, it has oxygen. So P is correct. Now, what about R? R is the structure, the main body of the glycoprotein or the glycolipid. So, of course, it must contain oxygen. That's good. S, yeah, it will contain oxygen. That's fine. But the main part is about Q. Q is a fatty acid tail. Is it gonna have oxygen? It's not gonna have oxygen. So it's not B either because that has Q. So it's P, R, and S only. And this is why the answer is C. Question number 19. Adrenaline is a hormone that prepares the body for rapid responses by increasing the blood flow to the muscles and increasing the blood sugar concentrations. Why does that happen? Why do we increase the blood sugar concentrations? It's because we need to ensure that all of the cells take in as much sugar from the blood as they need, and they can use the sugar, or in other words, glucose, break it down to release energy, which they can use for their flight or fight response. Now, adrenaline is secreted from the adrenal glands. This is the hormone. And it acts on nearly all body tissues by binding to adrenergic receptors on the surface of the cells. Now, different tissues respond to the adrenaline hormone in different ways. So, for example, if you look at the smooth muscle cells in the airways, adrenaline causes them to relax. But if you look at the smooth muscle cells in the arterioles, adrenaline causes them to contract. What could explain the different responses of the smooth muscle cells to adrenaline? So they're asking you, why is it that in the airways the same smooth muscle cells relax, but in the arterioles they contract? Let's see. Now, we know one thing for sure. It's the same hormone that is being used, right? Adrenaline. It's the same ligand which is being used. So if any answer choice uses the word different ligands, it's going to be wrong, which is exactly what option B says. The adrenergic receptors on the smooth muscle cells in the airways bind to different ligands when you compare these to the ligands which bind to the adrenergic receptors on the smooth muscle cells in the arterioles. That's wrong. It's the same ligand, which is adrenaline. Ligand is basically that thing or that structure or that molecule which binds to the receptor. So it's the same ligand. So B is wrong. Now, the concentration of adrenaline reaching the target cells is dependent upon the distance from the adrenal glands, and it determines whether the smooth muscle cells relaxes or contracts. This is BS. Why? This is basically telling us that if you are closer to the adrenal gland, that means more adrenaline is going to reach to you, right? Closer to the adrenal gland, more adrenaline is, I'm having a hard time saying it, is going to reach to you. When there is more adrenaline, the smooth muscle is going to contract. But if it's less adrenaline, the smooth muscle is going to relax. How does that make sense? Imagine if the airways were further, or imagine if the airways were closer to the adrenal gland. Then that would have meant that there would have been more adrenaline reaching to the airways. So does that mean that the airways would have contracted and you would have choked? Of course not. This is wrong. C is wrong. Okay, this doesn't make sense. Now, the binding sites of the adrenergic receptors, okay, now we're talking, the binding sites of the adrenergic receptors on the smooth muscle cells have different tertiary structures. Nah, lost me there. Absolutely not. Why? Let's understand. Adrenergic receptors binding sites, basically the place where the adrenaline, the ligand is gonna bind, it's saying that the binding site have different tertiary structures. If it has different tertiary structures, how is the same hormone binding to both of them? So that's wrong. That leaves A with our answer, that smooth muscle cells in the airways and in the arterioles have different types of adrenergic receptors. That could happen too, right? If different receptors, then different signal transduction pathways. If different signal transduction pathways, then different signaling cascades. And when you have different signaling cascades, that's when you have different responses. And that is exactly what we were told here, that in one scenario, the smooth muscle cells contract and in the other, they relax. That's a different response each time. How does that happen? Because different signaling cascades. How does that happen? Because different signal transduction pathway. These two words which I'm constantly repeating, signal transduction pathway and signaling cascade, these are the two processes or the two steps in the process of cell signaling. So make sure you guys read through cell signaling one more time if you're not sure of what I was talking about. Okay, question 20. What explains the effect of, or what explains the effect on a red blood cell if being placed into pure water? Think about it. You have a container, You have pure water. Okay, by the way, what's the water potential of pure water? Half of the students don't know this. The water potential of pure water is 0 pascal or 0 kilopascal. Why is that? Water potential can never be positive. You can never have something which has a water potential of +3 or +47 or +67. It kilopascal. It's not possible. So how do you know that? Look at this. Pure water. Pure water means it is water that is free of any impurities. It's this is the thing, right, which is gonna have the maximum amount of water in it. Anything else apart from pure water must have a lower water potential than it, right? Now, pure water itself has a water potential of 0. So if we are saying that anything else is going to have a water potential lower than this, now that means all of the liquids, all of the solutions apart from pure water are going to have negative water potentials, -3 kilopascals, -7 kilopascals, -23 kilopascals. And the symbol for water potential is psi or phi. How do you draw it? Draw U and a stick in between like this. Now, think of a number line here. So if you go to the right, the answer gets more positive. If you go to the left, the answer gets more negative. This is 0. Let's say -3, -7, -10. This is maximum water potential. Think about it. Which one is going to have a higher water potential, -3 or -7? You would say -3 because it's closer to 0, right? It's closer to the number which pure water has. So that's just another way to remember. The low negative or the smaller negative the number is. So let's say -3 and -23. Which number is less negative? -3. Or you could say which number is more positive? -3. That's going to have a greater water potential. But anyways, this wasn't the question. Let's see what the question is. What happens to the red blood cell if you place it in pure water? Pure water, of course, has a higher water potential if you compare it to the water potential of the cytoplasm of the red blood cell. So the surroundings is going to be higher water potential, inside is going to be lower. So how does osmosis happen? What is the definition of osmosis? It is the movement of water from a region of high water potential to a region of low water potential down the water potential gradient through a semi-permeable membrane. So the water is going to go inside the red blood cell. So let's look at the answer options. Less water leaves the cell than enters it. That's right. So the cell swells and bursts. That's correct. Why does the cell burst though? Because it's a red blood cell, right? It's an animal cell which does not have a cell membrane. So it does burst. But let's see the other options too, just for extra safety. More water enters the cell than leaves it. Correct. So the cell becomes turgid. That's right. But what happens after it becomes turgid? It bursts. So not the entire thing as mentioned here, has been mentioned here. C. Water can enter the cell, but water is unable to leave the cell. No, 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 no. Now, this is important. When you talk about the movement of water, and when I say osmosis is from outside the cell to inside the cell, it does not mean that this water movement is only one way. No. What's actually happening in real life? In real life, let's say you have 10 water molecules here and you have 3 water molecules here. In real life, let's say 6 water molecules move in, and then at the same time, only 1 water molecule or 2 move out. How many moved in? 6. But how many moved out? 2. What's the net movement? What's the overall movement? 4 inwards. So when you talk about osmosis, osmosis is not a one way movement. This is actually explaining the overall movement. So when we say osmosis is the water moving into the cell, the water is not just moving into the cell. You will say that the water is overall moving into the cell, meaning there is still some water that moves out, but that's much lesser compared to the amount that moves in. So this is wrong when we say that the water is unable to leave. No, it does leave the cell, but it's much lesser compared to what enters it. Now, water enters the cell at the same rate as at which it leaves the cell. No, in that case, then the net movement would be zero, right? This is what we call an isotonic environment wherein the osmosis is zero. Think about when this could happen. When the water potential outside and inside is going to be the same. Is there any water potential gradient now? If the inside and the outside have the same water potential? No. So is the water is still going to move? Some water is going to move in, some is going to move out. But the net movement is going to be zero. So let's say if two water molecules move in, at the same time, two water molecules are going to move out. That's an isotonic environment. So of course the cell shape will change. It's not that it will not change. So A is the answer right here.